Hello, welcome. Welcome to my channel Deep Learning Biology. My name is Dr. Vikash Bhardwaj. Now, before starting the video, I will request you have one glass water. Have one glass water. Remember, keep in mind, water is going to boost your memory. You are 70% water. Try to think here. Try to think here. In the whole universe, you will not see chemical like water. Just try to have water. Marvelous properties. Just drink one glass water. Okay, this water is going to boost your memory. Now, here I just want to tell you, meet my student Homo. Homo, here I just want to tell you, in his family, okay, his father used to run many companies, majorly biotechnology companies. In future, Homo has to take care of all these companies. He has to run family business. Okay, that's why he is learning fundamentals of biology. Now, I have another student, Sapiens. Sapiens, she is not from a rich family, okay, but she want to be a doctor, she want to be a scientist, okay. So, she is working, she is learning things in a focused way so that she can achieve her dreams, okay. Yes, in future, she might want Nobel Prize, she may start any big companies, okay. Now you know very well, most of the Nobel laureates, okay, they actually runs big companies, okay. So these are two my students. One name is Homo, another name is Sapiens. Yes, I know also your scientific name is Homo sapiens, so don't get confused. Now here I just want to tell you one of the good characteristics, you can say, of Homo sapiens is that they eat well, they play well, they sleep well. They eat well, they play well, they sleep well. Why? Because they know they have to study. Their primary focus is education. Okay? So they maintain one thing, they are eating well, they are playing well, okay, they are sleeping well. They maintain healthy sleep cycle. Okay, so today Homo sapien, they are going to learn about protein secondary structure. Okay, so now the Homo sapien have the deep breath. Just control your breath. Okay, just control, have deep breath. Slow down your breath. This is important. Remember, in life, you need confidence. What you need? Confidence. Not so much of confidence. You need little bit of confidence to do great things. You need just confidence. Okay? So with me, you are going to build confidence. Now, be ready and start learning. Today, I am going to teach you the secondary structure of proteins. Secondary structure of proteins. Okay? Now, let's move forward. So, just focus your mind. Okay? Just focus your mind. Be attentive. And today, I am going to teach you proteins secondary structure. Now, here, I just want to tell you that in the previous class, yes, I know I have to clear the doubt, okay. So, in the one lecture, I have already told about proteins primary structure. Now, in this class, you are going to learn about proteins secondary structure. So, here, Homo sapiens learn clearly proteins secondary structure. Just I want to remind you about the primary structure of the proteins. Here, hopefully you remember, the primary structure of the proteins, it actually provide you information related to the sequence. You know very well, in a protein, what you are going to have. Proteins are polymer of amino acids. So, primary structure of the proteins, it is going to tell you what kind of amino acid, what types of amino acids are present within the proteins. It is going to tell you the sequence, okay, of the amino acids which are present within the proteins. Primary structure also tell you all the covalent bonds, peptide bonds, disulfide linkages which are present within the proteins. So here, if you see, this is the structure of insulin. Insulin, it is going to have two chains. Okay, so here you are going to have two chains. You can see the amino group, the carboxylic group, amino group, carboxylic group of both of the chains, and here you can see the single letter code. So keep in mind, the primary structure of the protein, it actually tell you the what kind of amino acids they are present within the protein. It is going to tell you the sequence information. Yes, here you can also see the peptide bonds, disulfide linkages. Okay, so this is about the primary structure of the proteins. Clear? Now let us move forward. Now here, if you see, in tertiary structure, we will discuss the overall 3D structure of the protein. In tertiary structure, we will discuss the overall 3D structure of the protein. Just look this structure, the 3D structure. Okay, you can't explain the 3D structure in one go. Okay, so what scientists they generally do, they study the secondary structure of the proteins. So in secondary structure, keep in mind, we are going to learn about local spatial arrangements within the protein. So you see, a protein, okay, is going to have, if it is a one single polypeptide, it is going to have 3D structure. Okay, now this 3D structure we are going to study in chunks, we are going to study in parts, and here in secondary structure, what we are going to study? Local spatial arrangements. Now, here you see, this is one local spatial arrangement known as alpha helix. Another is beta sheets. So today we are going to discuss about alpha helix, beta sheets, beta turns also in secondary structure. Now here, listen carefully, try to see here, in a protein, okay, what you are going to see? You are going to see amino acids. Proteins, it is going to have, proteins are actually the polymers of amino acids. Here amino acids, they have been joined. Now these amino acids, okay, they are going to form at some location regular patterns. So within the protein, what you are going to see? Regular patterns. You can say repeating local fold, repeating local structures. At some point, you are going to see random coils. So try to see, in a protein, what you are going to see? Regular patterns, or you can say repeating local folds. At some points, you are going to see random coils. Now here today our focus is on regular patterns, the recurring structures or patterns, repeating structures. Try to see here, in a protein, these recurring structures of patterns, generally these are stabilized by hydrogen bonds. And here these recurring structure patterns, they are going to give rise to secondary structure. So here when we are going to learn about the secondary structure of the protein, remember our focus is on repeating recurring structural patterns within the proteins. Hopefully everything clear. Now try to see here. You see Proteins are polymers of amino acids. Here, if you see the structure, let's take one dipeptide. Okay, so here I have shown you one amino acid, another amino acid. Okay, both have been joined, and here in between you are going to see formation of peptide linkage. This is peptide linkage, peptide bond. Okay, now here in a protein you are going to have chain of amino acids. Okay, one amino acid joined to another, another to another, another to another, another to another. Now here if you see the peptide bond, just focus on the peptide bond. Okay, C double bond O. Okay, now this double bond O, okay, due to the phenomena of resonance, okay, this double bond O, it will be spreaded over the peptide bond. Okay, so here you see the peptide bond. Okay, this is going to have double bond character, partial double bond character due to resonance. Okay, now here when you are going to have double bond, okay, so here you see C and N, in between C and N you are going to see single bond, but this double bond between C and O, okay, it will also show its effects on C and N. So here you see the peptide bond is also going to carry the partial double bond character. When you are having single bond, okay, it can move freely, but when you are having partial double bond, okay, it will not move freely. It is going to become rigid. Yes, all these atoms which are present within the peptide bonds, they are planar, means they are going to be present in a single plane. Now here if you see the peptide bond, it is going to carry partial double bond character. It is rigid, it is not flexible so much, but there is some flexibility. At some points you are going to see the bond, they can rotate. Now try to see here. Here you see I have not shown the single double bond O. Here single bond O. Okay, so here if you see C N C alpha C, this is going to represent the peptide bond. Now here you see between N and C alpha you are going to have angle. This angle is known as phi angle. Okay, same way between C and C alpha, okay, the atoms, they are going to have some position and they are going to make an angle. Here it is phi. Okay, so they are having some rigidity. Okay, they are not just moving freely. Okay, so here they are going to make some angle. Now these angles, these are known as dihedral angles. Phi and psi, keep in mind these are dihedral angle. So between N C alpha, okay, so this is phi, and C alpha C, it is, okay, psi. Okay, now keep in mind here the rotation is permitted, even though it is rigid, but rotation is permitted. Okay, now when this, okay, protein, let's suppose this peptide, it is fully extended, fully stretched. Okay, so in this situation what will be the angle? Very simple, 180 degree. Okay, but keep in mind, okay, so this is not going to be possible in natural protein, but you can think if you are going to stretch completely, okay, it will be in linear form, and in this situation what you are going to have, phi and psi value 180 degree. Now here if you see NC alpha, it is going to represent phi, C alpha C, it is going to represent psi. Okay, so at these, okay, so it is going to form two angles, okay, due to some, okay, flexibility. Okay, now here the rotation of bond is permitted. Okay, and here if you see the value of these angles, it may be from minus 180 to plus 180. Okay, so within the proteins, okay, one amino acid is there, there is another amino acid there, okay, they are going to have different phi and psi angles. Okay, so the phi and psi angles, they help us a lot in finally concluding the secondary structure of protein. Here keep in mind, if phi and psi is 0, okay, generally it is a theoretical situation, it can't be possible, okay, just we take it as a reference point, okay. Generally this situation is prohibited in proteins. Okay, so we generally start with phi and psi 0, then we start what is the angle. Okay, it can lie in between minus 180 to plus 180, minus 180 to plus 180, and generally we are going to plot a graph, and that graph is known as Ramchandran plot. Okay, so scientists generally what they are going to determine, they are going to determine phi and psi angle, and they are going to plot a graph. That graph is known as Ramchandran plot. Now keep in mind, Ramchandran plot, if you are going to see Ramchandran plot, it will tell you what the angles are allowed, okay, what the angles are allowed, what the angles are not allowed. Okay, so by studying, okay, the angles phi and psi within the proteins, okay, by using Ramchandran plot, okay, scientists, they can study the secondary structures which are present within the proteins, and they can study what angles are allowed, what angles are not allowed. Okay, so here you see it started from 0, 0, then from minus 180 to 1 plus 180, and this way you are going to plot the graph, and here you can see different regions. Okay, so in one particular type of helix you will see the angle will lie in this region, in another type of helix you will see the angles they lies in that region. Okay, so Ramchandran plot allowed to see the allowed conformations. Now here if you see the secondary structure of the proteins, okay, yes, you can study by using Ramchandran plot, and here if you see in the case of secondary structure of proteins, what we have to see, the repeating structural patterns, local repeating structural patterns which are generally found within the proteins. So here keep in mind, the local repeating structural patterns which can generally be found within the proteins are alpha helix, second one is beta sheets, and the third one is beta turns. So Homo sapien, today we are going to learn about alpha helix, beta sheets, and beta turns. Now try to see here. In the case of alpha helix, here the best example is hair protein. Just okay, remember this. So here within the hair, most of the proteins, they are going to align, they are going to have the super secondary structure in form of alpha helix, and you know very well in hair protein it is alpha keratin. Here the regular repeating pattern is alpha helix. So in case of alpha keratin, the regular repeating pattern is alpha helix. Okay, now here what you are going to see, one helix. You are going to see one helix. Here the polypeptide backbone, if you are going to see, it is wrapped around one axis. So there is one imaginary axis. Around this, the polypeptide, it is wrapped around, and it is going to form alpha helix. Now try to see here. In the case of alpha helix, remember, okay, so here what you are going to see, the R group which are present, okay, on amino acids. Keep in mind the R group, they will be present outside of the imaginary axis. And if you see each turn, so here you are going to have helix, okay, it is going to have turn, and each turn of the helix, it is going to have 3.6 amino acid residue. It will have 3.6 amino acid residue, and length of this will be 5.4 angstrom. Okay, this is very important, keep in mind. Next here, if you see the alpha helix, okay, the ideal angle of phi and psi for alpha helix are minus 57 degree, phi, psi minus 47 degree. Phi minus 57 degree, psi minus 47 degree. And here keep in mind the alpha helix, they can be right-handed or left-handed. There can be both right-handed as well as left-handed. In case of right-handed, try to see, here you are going to have turns. Okay, in case of right-handed, okay, these turns, they will move towards right hand. In left-handed, the turns, they are going to move towards left hand. So you can have both right-handed alpha helix and left-handed alpha helix. Now try to see here. In alpha helix, okay, what you are going to see, that this whole complete helix, it is being stabilized by hydrogen bonds. So here you are going to have peptide bonds, but the whole helix, it is being stabilized by hydrogen bonding. And keep in mind here, every peptide bond is participating in hydrogen bond formation. Here the first nitrogen of the, let's suppose the first peptide bond, first nitrogen of the first peptide bond is going to make hydrogen bond with the fourth amino group of the peptide bond. So there is hydrogen bond between first nitrogen of the peptide bond and the fourth amino of the peptide bond. Hopefully you can see in the structure, okay, there is formation of hydrogen bond between first nitrogen of the peptide bond and the fourth amino of the peptide bond. So here keep in mind the alpha helix, they are mainly stabilized by hydrogen bonding. Now here if you see each turn of the alpha helix, so you are going to see the turn and you are going to have adjacent turn. So there is one turn, then another adjacent turn. Okay, here in between them you can see there are total 3 to 4 hydrogen bonds. So hopefully in this picture you can see clearly. Okay, in between adjacent turns, what you are going to see? 3 to 4 hydrogen bonds. So here I have told you that the alpha helix, they are being stabilized by hydrogen bondings. Hopefully all these points are clear to you, Homo sapiens. Now let us move further. Try to see here. If you see in proteins, generally 1/4 of the amino acids, they are going to form alpha helixes. Okay, so if you are going to have any protein, yes, you are going to see this supersecondary structure, alpha helix. But also keep in mind that in not all the polypeptides, they can form stable alpha helix. Not all the polypeptides can form stable alpha helix. Try to see here. Let's suppose you are going to have a polypeptide. Okay, in that polypeptide, let's suppose you are going to have long chain of glutamic acid. Okay, so you are going to have amino acid sequence. At one particular location, you are going to have glutamic acid, glutamic acid, glutamic acid, glutamic acid, glutamic acid, glutamic acid. So you are going to have chain. Now try to recall. Glutamic acid is negatively charged amino acid. So wherever you are going to have the stretch of glutamic acid, the protein, it will carry negative charge. Okay, so wherever you are going to have stretch of glutamic acid, what you are going to have? The you are going to have negative charge. So this will prevent formation of alpha helix. So in alpha helix, where you are going to have glutamic stretch, okay, that is not going to form stable alpha helix. Okay, so that is not going to form the stable structure. Okay, so here keep in mind, not in all the proteins you are going to see stable helix. So one condition is this. Yes, if you take another situation, in polypeptide, if you are going to have the situation like, let's suppose you are going to have lysine and arginine, and let's suppose you are going to have many lysine and arginine. Now you know very well, lysine, arginine, histidine, these three are positively charged amino acid. So when you are going to have stretch of lysine and arginine, what you are going to have? Positive charge. So here there will be repulsion between positive. Okay, so here when you are going to have a stretch of lysine, arginine, this will also prevent formation of alpha helix. This will also prevent formation of stable alpha helix. Clear? Now let's move further. Okay, so these are some of the basic important point you must remember. Okay, now let's see here. In the situation, if you are going to have proline, if you are going to have glycine within the alpha helix, okay, then the presence of proline and glycine is going to destabilize the alpha helix. Okay, you will not see the formation of alpha helix. Try to see here. If you see the case of proline, glycine is too small. Proline, if you see, it is going to have rigid ring. And here, if you see, if you remember, in case of proline is amino acid. Here, the rotation about NC alpha not allowed, not possible. Okay, if you just look into the structure of proline, rotation about NC alpha, it is not possible. So that's why in alpha helix, what you are going to see, the presence of proline. Actually, the proline is going to introduce destabilizing kinks in alpha helix. So this is going to destabilize the alpha helix. So naturally, in proteins, wherever you are going to see presence of alpha helix, you will not see the presence of proline. You will not see the presence of glycine. So here keep in mind, okay, proline and glycine, they are rarely found in alpha helix. Very important. Proline and glycine, they are rarely found in alpha helixes. Clear? Now let's move forward. Now try to see here. We are now going to discuss another structure, beta conformation. Beta conformation. Here, in the case of beta conformation, you are going to see zigzag pattern. What you are going to see? Zigzag pattern. Try to recall, in alpha helix, what you are going to see? You are going to see a coil. Here in beta conformation, what you are going to see? Zigzag conformation. Okay, so this is zigzag conformation showing beta strands. Here a single protein segment is present in form of beta conformation. Now here, if you are going to have, okay, many side by side arrangement of beta strands, okay, so what you are going to have? Many beta strands, and they are lying together, and they are, okay, either be parallel or anti-parallel. What they are going to form? Beta sheets. Okay, side by side arrangement of beta strands, it will lead to formation of beta sheets, or you can say beta pleated sheets. Now try to see here. You can see this is the structure of beta pleated sheets. Here the beta pleated sheets, in between beta pleated sheets, what you are going to see? Hydrogen bonds. Here also you see the hydrogen bonds, they are going to stabilize beta pleated sheets. Now try to see here what you can have. these strands, they either may run in parallel direction. Okay, you can have parallel beta sheets. So when all the strands they are running in parallel direction, what you are going to see? Parallel beta sheets. Now here in case of parallel beta sheets, remember blue color, these are representing the hydrogen bonds. Here the hydrogen bonds are distorted. They are not in line. So keep in mind, in parallel beta sheets, the hydrogen bonds they are distorted. And if you see the case of anti-parallel beta sheets, so here you can see one chain, it is moving in different direction. Two chains, they are running in one direction. So here what you are going to see, formation of anti-parallel beta sheets. Anti-parallel beta sheets. And here if you see in the case of anti-parallel beta sheets, what you are going to see? In-line hydrogen bonds. The hydrogen bonds are in line. Okay, so hopefully now it is clear the difference between parallel beta sheets and the anti-parallel beta sheets. Now, if you see, normally in case of proteins, keep in mind what you are going to see. Generally, you are going to see anti-parallel beta sheets twice more. Okay, you can say more common is anti-parallel beta sheets compared to parallel beta sheets. Okay, so in a protein, generally what you are going to see? Anti-parallel beta sheets. So here beta sheets, they can be parallel and anti-parallel, and generally what you are going to see? Anti-parallel beta sheets. Now keep in mind the example of beta sheets, it is silk. Okay, the silk protein. And here if you see the silk, it is going to have high tensile strength. Why? Because here the proteins, they are being arranged in the form of beta sheets, and in between beta sheets what you are going to have? Hydrogen bonds. Clear? Now let us move Homo sapiens. Now here we are going to discuss beta turns. Okay, try to think about a globular protein. If you are going to see any globular protein, in the protein you are going to have alpha helices, you are going to have beta strands. Okay, so when the protein is going to fold at some points, okay, what is going to happen? The polypeptide is going to reverse its direction. Okay, so there is a protein, it is folding. At some point, it is going to reverse its direction. Okay, so at these points where the polypeptide is going to reverse its direction, and generally it is going to reverse its direction by 180 degree. At that location, you are going to see there will be 4 amino acids. Actually, they are going to form beta turns. So here keep in mind, within the beta turns, you are going to have 4 amino acids, and what they are going to represent? Okay, they are going to represent a complete turn, 180 degree turn. Now here you can see in this picture, okay, so here you can see in yellow color, okay, the 2 chains, and then above you can see the beta turns. Okay, so just they are connecting the strands. Okay, so here keep in mind the beta turns, they are going to represent, they are going to be they are present where you are going to have something 180 degree turn. Now here hopefully you can see the presence of beta turns. Now here I just want to tell you, in case of beta turns, I have told you, you are going to have 4 amino acid. Okay, you are going to have 4 amino acid. Keep in mind, between the first amino acid and the 4th amino acid, you are going to see hydrogen bonds. So here in case of beta turns, between first amino acid and the 4th amino acid, what is there? There is presence of hydrogen bonds. Okay, Another important point, keep in mind, in beta turns, what we are going to see, the presence of glycine and proline. Hopefully you remember, glycine and proline, they are generally absent in alpha helix. Okay, but here in beta turns, what we are going to see, the presence of glycine and proline. Remember, glycine and proline, they are common in beta turns. Now, here, if you see the secondary structure of the proteins, they can be studied easily by using a special type of spectroscopy, that is circular dichroism spectroscopy. Circular dichroism spectroscopy. Okay, here scientists, what they used polarized light, and then they study the CD spectra. And here, for different types of secondary structures, okay, you are going to have different types of CD spectra. Okay, so by studying the CD spectra, the scientists, they can predict what percentage of alpha helix are present within the protein, what percentage is beta conformation, okay, what percentage is unstructured, random. Okay, so by using circular dichroism spectroscopy, the scientists, they can study the secondary structure of proteins. Now, here you can also study, you try to see the proteins. If you are going to heat, okay, what will happen? They will come in unfolded state. Okay, so protein folding you can study by circular dichroism spectroscopy. Okay, you can study, okay, what portion of the protein is folded, unfolded, by studying the CD spectra. So, hopefully, Homo sapien, you have learned clearly about secondary structure of the proteins. Thank you. Thank you for watching this. If you have any doubt, leave the comments. And keep in mind, before going for sleep, Homo sapien, this is very important. You must build a strong mind. This is the need. You have to build a strong mind, and whatever you have learned now, it must go in your permanent memory. Permanent memory. How it can go? When you are going to revise the things. Just don't watch now any video game, any other type of funny type videos. Okay? Here, just revise your notes. Revise. If possible, try to watch the video again. You can do. Okay? You will learn the things more better way if you have missed something. Remember, more the practice you are going to do, the things they will move in your permanent memory. So here, before going for sleep, okay, just try to recall what you have learned today. Your primary focus must be in learning, and if you have learned this, you can teach well, far better than me. Far better than me. It's okay. So whatever, try to learn things and try to build the confidence. If by chance you can have discussion with your friends, you can discuss, that is also better. Okay, so here just try to build the confidence. Okay, whatever you want to achieve, you can achieve easily, just you need little bit of confidence. Now relax yourself. Okay, thank you for watching this. Bye bye. Take care.